students. Today I am going to present the topic entitled Organization of the Human Genome. The objective of this lecture is to talk about the architecture and organization of the human genome, outlining the different classes of DNA sequence and briefly describe their function. The human genome is composed of two parts, a nuclear genome and a mitochondrial genome. The nuclear genome consists more than 26,000 genes and the mitochondrial genome consists of only 37 genes. The nuclear genome provides the bulk of genetic information and is distributed in either 23 or 24 different types of chromosomes. That is, 22 autosomes plus an X chromosome in females and an additional Y chromosome in males. The mitochondrial genome is a small circular DNA which encodes some components needed for mitochondrial protein synthesis. However, the nuclear genes encode the most of mitochondrial proteins which are synthesized in the cytoplasm and then imported into mitochondria. It is presumed that only 5% of the human genome is functionally important and has been strongly conserved during evolution, out of which only 1.1% of the human genome contains the protein coding DNA sequences and 4% consists of the non-protein coding DNA sequences. These non-protein coding DNA sequences are strongly conserved, which include functionally important RNA molecules and many cis-acting sequences that regulate the gene expression. Now we shall talk about general organization of the human genome. The human mitochondrial genome sequence was published in 1981. Which has helped us to understand the working of mitochondrial DNA. However, the nuclear genome posed a formidable challenge and its sequencing began in 1990s, and by 2004 the entire euchromatic portion of the genome was sequenced. Now we'll talk about the mitochondrial genome. The human mitochondrial genome is composed of a single circular double-stranded DNA which is 16.6 kilobases in length. The two mitochondrial DNA strands have different base composition. The heavy H strand is rich in guanines, but the light L strand is rich in cytosines, and overall base composition of the two mitochondrial strands is 44% G plus C. There are thousands of copies of the double-stranded mitochondrial DNA molecules in a cell, but its number varies in different cell types. There are 37 genes in the human mitochondrial genome, and out of which 28 genes are encoded by the H strand and 9 are encoded by the L strand. About 2/3, that is 24 out of 37 of the mitochondrial genes specify functional non-coding RNA as their final product. Out of these 24 mitochondrial genes, 22 transfer RNA genes make 22 types of mitochondrial transfer RNA and 2 ribosomal RNA genes make 16S ribosomal RNA and 12S ribosomal RNA. These are the components of mitochondrial ribosomes. The remaining 13 genes encode polypeptides which are part of the mitochondrial respiratory complexes, that is, the enzymes of oxidative phosphorylation engaged in the production of ATP. However, the majority of the polypeptides of the oxidative phosphorylation system and other mitochondrial proteins are encoded by the nuclear genes which are translated in cytoplasm before being imported into the mitochondria. The human mitochondrial genome is compact and all its genes lack introns. The coding sequences of some genes show some overlap, and in most other cases, the coding sequences of neighboring genes are contiguous or separated by one or two non-coding bases. Some genes lack the termination codons, and this deficiency is overcome post-transcriptionally by the addition of UAA, that is the stop codon. Now we shall talk about the nuclear genome. The size of the human nuclear genome is 3.1 GB, that is 3100 MB, which is distributed between 24 different types of chromosomes comprised of double-stranded DNA molecules having the histones and non-histone proteins bound to it. There are 22 types of autosomes and 2 X chromosomes, X and Y, which can easily be differentiated by chromosome banding. These chromosomes have been classified into groups according to size and the centromere position. The nuclear genome size is 186,000 times that of mitochondrial DNA molecule. However, the nucleus contains more than 99% of the DNA of the cell. The focus of Human Genome Project was primarily on sequencing the euchromatin portion. It is the gene-rich region of the nuclear genome amounting to 2.9 GB, and it is transcriptionally active. The remaining 200 MB of the genome is heterochromatin region, which is permanently condensed and transcriptionally inactive. The heterochromatin region is composed of highly repetitive DNA that is difficult to sequence. The chromosomal DNA varies in length, and the proportions of euchromatin and heterochromatin in them. There is some heterochromatin in each chromosome at the centromere, and in the chromosome numbers 1, 9, 16 and 19, the heterochromatin is present in the euchromatin region close to the centromere, that is pericentromere, and in acrocentric chromosomes there are 2 heterochromatin regions. In the Y chromosome, most of the DNA is organized as heterochromatin. In human genome, the euchromatin component has an average base composition of 41% guanine plus cytosine. However, there is a variation in different chromosomes. Example, 38% guanine plus cytosine for chromosome numbers 4 and 13, and up to 49% guanine plus cytosine for chromosome number 19. The base composition percentage also varies along the length of chromosomes. Now we shall talk about the CPG Islands. The human nuclear genome has an overall lower frequency of dinucleotide CPG. However, there are some stretches of unmethylated or hypomethylated DNA with normal CPG frequency. Such islands of normal CPG density are comparatively GC rich, typically more than 50% guanine plus cytosine, and extend over hundreds of nucleotides. CPG islands are gene markers because they are associated with transcriptionally active regions. Highly methylated DNA regions are prone to adopting a condensed chromatin conformation. But for transcribing DNA actively, the chromatin needs to be in a more extended and open unmethylated conformation that shall allow various regulatory proteins to bind more readily to promoters and other gene control regions. The nucleotide CPG DNA is a common target of cytosine methylation to form mCpG by cytosine methyltransferases. DNA methylation has an important consequences for gene expression and allows particular gene expression patterns to be stably transmitted to daughter cells. It has also been implicated in systems of host defense against transposons. Cytosine methylation is highest in vertebrates among the animal kingdom, and it is dispersed throughout the genome, and in human DNA only 3% of cytosines are methylated. Now we shall talk about the uneven distribution of genes on nuclear DNA. There is an absence of genes on the heterochromatin region, and the density of genes on the euchromatin genome varies along the chromosomes. The insight on the distribution of genes was obtained for the first time in a metaphase chromosome. It was later on found that some chromosomes are gene-rich. For example, 19 and chromosome 22, and some are gene-poor. For example, chromosome 18. This difference in gene density can also be seen with Giemsa staining, known as G-banding of chromosomes. Regions with a low guanine plus cytosine content correlate with the darkest G bands, and those with a high guanine plus cytosine content with pale bands. However, the predictions of differential CPG island density and differential gene density were subsequently confirmed by analyzing the human genome sequence. Now we shall talk about RNA encoding genes. At the start of the Human Genome Consortium, it predicted that there were 30,000 protein coding genes. The validation of the gene prediction by evolutionary comparisons with other mammalian genomes failed to find the counterparts of many human genes. By the late 2009, the protein coding genes appeared to be stabilized somewhere around 20,000 to 21,000, but there remained the uncertainty about the human RNA genes. This uncertainty is because the computer program analysis of the genomic sequences for RNA genes is difficult, and the reasons are the absence of reading frames and smaller size of some RNA genes which have the evolutionary conservation. There are about 6,000 human RNA genes, which also include the genes which encode the non-coding RNAs, which are thought to be important for the gene regulation. The human genome contains at least 26,000 genes, but the exact number is difficult to determine. The combination of about 20,000 protein coding genes and at least 6,000 RNA genes gives a total number of at least 26,000 human genes. This remains a provisional total gene number because defining the RNA genes is challenging, and it will be some time before we can obtain an accurate human gene number. Besides, there are also tens of thousands of tiny human RNAs, and most of them are produced by the processing of single RNA transcripts. Now we'll talk about repetitive DNA. The bacterial and mitochondrial genomes being small and are tightly packed than the nuclear genome of the eukaryotes. The repetitive DNA is the feature of large genomes, and they are important as well as present in abundance. These repetitive DNA are short non-coding sequences present in few to millions of copies. But some large or long repetitive DNA contains genes or parts of the genes, and these sequences are prone to genetic mechanisms that result in a copy number variation between different haplotypes. The copy number variation is extensive in human genome and it generates structural variation. These variations are being identified with the personal genome sequencing, and these will be helpful in identification and prevention of diseases. The repeated duplication of genes containing sequences gives rise to a gene family, and many human genes are members of multi-gene families which vary in copy number and distribution. Now we shall talk about repetitive sequences. In the non-coding DNA of genes, the highly repetitive DNA sequences are found in introns and flanking sequences. In addition, they are also found in the exons in different extents. There is a tandem repetition of very short oligonucleotide sequences comprising of 1 to 4 base pairs frequently, and the tandem repetition of sequences encoding protein domains is also common. Example, lipoprotein LPA encoded by the LPA gene on chromosome 6 contains multiple tandemly repeated Kringle domains, 15 or more in number, of about 114 amino acids long. The different Kringle domains show sequence identity at the DNA and in amino acid level. This property of the sequence identity at DNA sequence makes them prone to unequal crossover, and as a result, the LPA gene is subject to length polymorphism. Even at the nucleotide sequence level, the DNA repeats that encode the Kringle domains show very high levels of sequence identity, making them prone to unequal crossover. Now we shall talk about overlapping genes and genes within genes. There is a high gene densities in simple genomes such as human mitochondria, Escherichia coli, and Saccharomyces cerevisiae, which are roughly 0.5, 1 and 2 kb in size, because they contain partly overlapping genes. However, in humans, the genes are much bigger and the overlapping of protein coding sequences is less. The gene density varies from chromosome to chromosome and also within the region of the same chromosome. The overlapping genes may be present in the chromosomal regions with high gene density. Example, the class 3rd region of the HLA complex contains partly overlapping genes and it has an average gene density of about 1 gene per 15 kb. The genome analysis have shown that about 9% of the human protein coding genes to be overlapping. The overlapping can be partial or within the larger gene. Some small protein coding genes are also present. The example is the NF1 neurofibromatosis type 1 gene contains 3 internal genes. Further, it has also been seen that some RNA genes can also overlap with the protein coding genes. In some cases, few hundred nucleotides separate the two genes on the 5' end and the genes are transcribed in the opposite direction, regulated by a common promoter. Further, Some genes with common promoter are also transcribed in the same direction to produce a multigenic transcripts which is later cleaved to produce separate transcripts of each gene. This is called as polycistronic transcription unit, which is common in bacteria and mitochondrial genome. Besides, the nuclear genome also codes for some proteins produced by a common transcription unit. Example, the insulin chain A and B and somatostatin. Now we'll talk about gene families. The different classes of human gene families can be recognized on the basis of degree of sequence and structural similarity of their protein products. Genes operating in the same functional pathways but producing proteins with little sequence similarity are distantly related in evolution, and they are dispersed in different locations on the chromosomes. Example, genes encoding insulin and insulin receptors. These genes might have arisen from ancient gene duplication events, which may initially have been clustered together, but could have been separated by translocation or invergence. However, the genes producing proteins with both structural and functional similarities are often present in a gene clusters at a specific subchromosomal location, which may have arisen from recent gene duplication. Some gene families are organized in large clusters at different chromosomal locations. Example, olfactory receptor gene family, which encodes a diverse repertoire of receptors which allow humans to discriminate thousands of different odors. Now we'll talk about pseudogene. In addition to the functional genes, the gene families also have defective gene copies. The defective gene copy, which contains several exons of the functional gene, is called as the pseudogene, which arises from gene duplication events that produces two gene copies. One of the gene copies may pick up mutations, that is, genetic drift, to produce a pseudogene. Some defective gene copies having only little parts, a single exon of a gene sequence, is called as a gene fragment, that is, non-processed pseudogenes. These are often found in clustered gene families and have arisen by tandem duplication. Even though their sequences corresponding to the functional gene, they have inappropriate termination codons and aberrant splice junctions. Example, non-processed pseudogenes are found in an alpha-globin and beta-globin gene clusters. The pseudogenes are rare in prokaryotes. However, there are more than 8,000 different processed pseudogene copies of protein coding genes and more than 4,000 non-processed pseudogenes in the human genome. With this, we conclude today's topic. I hope you have understood.